Buenas tardes, bienvenidos a todos. Gracias por estar aquí y gracias a la gente que nos acompaña por Zoom y por YouTube. Realmente es un gusto presentarles el día de hoy a la doctora Judy Lieberman. Ella es física, obtuvo su grado en física de la Universidad de Harvard. Después hizo un doctorado también en física teórica por la Universidad de Rockefeller. Trabajó en física teórica en el Fermi, en el laboratorio de acelerador de Fermi en Italia. Y después cambió totalmente de rumbo su carrera. Obtuvo un grado en medicina por la Universidad de Harvard. Realizó su residencia en medicina interna en el Centro Médico de Nueva Inglaterra en Boston. Después hizo un postdoctorado en inmunología en el laboratorio del Dr. Hermann Eisen en el Centro de Investigación en Cáncer en el MIT, también en Estados Unidos. Y después trabajó también en la clínica en hematología y oncología en el Centro Médico de Tufts en Nueva Inglaterra. La doctora Lieberman ha recibido muchísimos reconocimientos, solo quiero mencionar algunos. Y ha ocupado diferentes posiciones de investigación. Desde el 96 está asociada a la Universidad de Harvard, donde empezó como profesora asistente. Después, a partir del 2004, ya se convirtió en profesor en el Departamento de Pediatría de la Universidad de Harvard también. Y ahorita, desde el 2012 al presente, ocupa una posición importante en el programa de medicina celular y molecular del Hospital Infantil de Boston. La doctora Lieberman, como mencionaba, ha recibido diferentes reconocimientos a su trayectoria académica y solamente quiero resaltar algunos de los que yo considero que son más importantes para no alargarme demasiado. Y es que desde el 2020 a la fecha fue nombrada miembro de la Academia Nacional de Medicina. En el mismo año también fue nombrada miembro de la Academia Nacional de Ciencias y en el 2021, el año antepasado, recibió el reconocimiento de investigadora distinguida en inmunología básica e inmunología tumoral. La doctora Lieberman ha hecho contribuciones muy importantes a la investigación, estudiando el papel de las proteínas que forman poros y la activación del sistema inmune innato, en estudiar las bases moleculares de la citotoxicidad mediada por linfocitos T citotóxicos y natural killers. También ha trabajado en diseño de terapias basados en interferencias de RNA. Y evidentemente, bueno, pues también tiene líneas de investigación muy relevantes en el papel de microRNAs pequeños y RNAs largos no codificantes en el desarrollo y en cáncer. Y me gustaría concluir simplemente resaltando que también la doctora Lieberman ha trabajado en diferentes enfermedades virales, infecciosas por virus, como HIV, el virus Zika, y también el virus herpes simplex que causa diferentes sarcomas. Quizás en los últimos años una de las contribuciones más relevantes fue el descubrimiento de cómo las proteínas conocidas como gasdermina se activan para inducir la muerte inflamatoria conocida como viroptosis y el papel que juegan esas moléculas no solamente en la respuesta inflamatoria en células del sistema inmune, sino en general en muchísimas células. Judy, thank you very much for being here. It's really nice to have you here sharing your knowledge with the students and with us today. And el título de su plática es cómo las células tumorales evitan el reconocimiento y su eliminación. Thank you, Judy.
Gracias. Gracias, buenas tardes. I'm sorry I can't give my lecture in Spanish, but it's been a pleasure being here, meeting the students and talking with the faculty. I'm going to give a talk today about the immune response to cancer. Some unpublished work, a lot of unpublished work. Let's see how it goes. So in the last decade or so, cancer therapy's really been revolutionized by the ability to manipulate the immune response to cancer, to awaken it and provide cures for some people with cancer. Immunotherapy is administered in two ways, by giving antibodies to inhibitors of T-cell activation, that's called checkpoint blockade, or by adaptively transferring engineered T-cells that their receptors, like with an antibody that will recognize a tumor coupled with a signaling molecule. And these therapies are generally extremely well tolerated. They have much less toxicity than the old style chemotherapy or even the targeted chemotherapy. And they basically cure cancer with very little evidence of drug resistance. However, the problem is that most tumors don't respond. And my philosophy has been that we have to, a lot of the efforts of people in the field have been to try to combine different checkpoint inhibitors to do better. They do a little better when they're combined, but they don't really make this therapy available to most people with cancer. And the idea that we've had is that you have to really go beyond. There are many ways that tumor cells avoid being recognized by the immune system, and you have to really try new strategies that might work with the therapies we have now, but really open up the field. So this is the way most cancer immunologists think about cancer immunology and immunotherapy. And it really focuses on the adaptive immune response. I'll just go over it. So in a tumor, there are always some dying cells, and those cells release antigens. Which are picked up by antigen-presenting cells, especially dendritic cells, that then migrate to lymph nodes, where they can present these tumor antigens to T cells, and mostly T cells. And those T cells get activated in the lymph node, and then they traffic through the bloodstream or the lymphatics, extravasate, and come back to the tumor where their T cell receptor can recognize the tumor cell and hopefully kill it. And the checkpoint therapy works, and also the CAR T cells work right here. They sort of release the brakes on T cell recognition and enable the cancer cell to kill. And then the killed cells can then cycle again through this cycle. So it's really a T-cell centric therapy that's only working at one part of the immune response to the tumor. Now, only tumors that have infiltrating lymphocytes respond to checkpoint inhibitors, but many tumors either don't express any antigens, they don't have infiltrating immune cells, and they're like an immune desert. Those tumors never respond to chemotherapy. Then there's another class of tumors in which there are antigens expressed, but for reasons that aren't actually that well understood, the immune cells especially the T cells, stay on the outside of the tumor and don't get in, aren't next to the tumor cells, so they can't help. And also, in a number of these tumors, there are very suppressive cells that really create an environment that makes it very difficult for immune cells to react. So it's only a small fraction of tumors that respond to checkpoint inhibitor. And in those tumors, the immune cells express inhibitory receptors, what we call checkpoint receptors, which the antibodies counteract and enable the T cells to function. So even in the tumors like melanoma or lung cancer that respond to immune therapy, only generally a minority of the tumors actually respond. So the question is, how can we do better? And so there's a concept in immunology called tumor editing. And I want to explain it to you because you'll need to understand it to understand my talk. So basically, the idea is when a tumor first forms, it is recognized by the immune system, but it's primarily recognized by what we call the innate immune system. Killer cells that can respond to common features of tumors, that can recognize the fact that a tumor cell isn't a normal cell. And when they recognize, and they're mostly natural killer cells, but also some kinds of specialized T cells like gamma delta T cells or NK T cells, which are sort of ancient types of T cells that can recognize features of the tumor. And when they recognize it, they can kill the tumor cell, and they secrete interferon gamma, which activates the antigen presenting cell in the tumor so that it can take up the antigens that are released from the dying tumor cell, migrate to the lymph nodes, and set up an immune reaction with T cells that could potentially recognize the tumor antigens. So initially, you get this immune response, which, until recently, when immunotherapy worked, people weren't really sure that this immune surveillance actually functions, except for tumors that were started by like viruses. But this is called immune surveillance. So the immune system can control the tumor. And in order for tumor cells to grow in an immune competent host like us, they have to figure out a way to evade this immune recognition. And so this immune surveillance network selects, it's like a kind of natural selection under the pressure of the immune system, selects for cells that avoid being recognized by the immune system. And that process is believed to occur early in tumor development in a new tumor, and it's called immune editing. And this theory was developed by Bob Schreiber about 20 years ago. Now, what's interesting is, because the tumors only appear once they've evaded the immune system, nobody has really systematically looked at how the tumor does it, how it evades the immune system, what genes get edited, how they get edited, and how that enables the tumor to survive. And instead, what cancer immunologists did after Bob described this process is they looked mostly at the T cell response to tumors, and they found that the tumors had downregulated the MHC molecules that present antigens. They had downregulated the mechanism for presenting antigens, but they didn't really look without bias at what was going on. And partly they didn't look because when the tumor is really small, it's not detected. So it only becomes detected after it's evaded the immune system. So as a consequence, we don't know a lot about immune editing. We know that that adaptive immunity is edited. Tumor antigens that might be recognized by the immune system are suppressed. And the machinery for presenting the antigen and processing it is repressed. But nobody has really looked at what other changes in the immune system. So to activate a T cell, to activate this adaptive immune response, which is a response to very specific proteins that are different in the tumor than in normal cells, you need three signals. You need an antigen, you need something called co-stimulation, and that's what checkpoint receptors block. But you need a third signal in order to make a really effective immune response and to make memory T cells, and that's a danger signal. And a danger signal basically tells the T cell that this isn't self, it's not a normal cell, it's something dangerous that I need to respond to. And that's the job of what we call innate immunity. It's basically a sign in the tumor that there's something wrong there. It's a pattern of a molecule that's typical of some kind of stress in the tumor that's different from normal cells. So people haven't studied at all innate immunity. And one of the reasons is that it's clear that some danger signals are also oncogenic. So if you smoke cigarettes or something, you turn on inflammatory pathways which promote tumorigenesis. So most cancer immunologists have been frightened about the idea of turning on these danger signals because they could be oncogenic, and they can. But the question is, once the tumor cell has already transformed itself from a normal cell to a cancer cell, that process has already occurred. And so that in order to really enlist the immune system, you may need to get the tumor to say, hey, I'm different, something you have to respond to. And as I said, there have never been any unbiased studies to see how the tumors evolve to evade immune recognition. And that's partly because once the tumor's evolved, it's already done that. So I just wanted to, so we're all on the same page. I know a lot of you are immunologists. Describe a little bit more about the innate immune system versus the adaptive immune system. So the innate immune system is made up of myeloid cells and a kind of lymphocyte called the natural killer cell, which can recognize common features of danger or damaged cells. And that's the immediate response to something that's changed, because it takes a week or two for what we call the adaptive immune response, which is made up of B lymphocytes that make antibodies, as well as T cells that can recognize the transformed cell and produce cytokines that will control it or recruit other cells to the area, and CD8 T cells, which can be cytotoxic and kill and eliminate the tumor. So cancer immunology has largely ignored this initial response. And in this cartoon, it suggests the innate immune response only occurs here at the beginning of the tumor, but actually it can occur all along here, because the tumor cells, unless they've been edited, should still look different to the immune system than normal cells. So innate immunity is the alarm system of the body, and it basically can distinguish self from non-self. It can distinguish bacteria that live on our skin and coexist with us, commensals, from pathogens. And it does it by recognizing either, for pathogens, molecules that are only produced by infectious organisms like lipopolysaccharide, or it can recognize what we call danger-associated molecular patterns, which would be the case in tumors. It's molecules that are present in sites which they wouldn't be. So for a tumor, if you had DNA in your cytosol, which some tumor cells have, that would be a danger signal that innate immunity could recognize. And there are special receptors in the cytosol which recognize, and in cancer, it's mostly mislocalized nucleic acids that are very important. So there are receptors that recognize RNA or DNA, RNA that's been modified or DNA in the cytosol that can be sensed as something dangerous. And the most inflammatory pathway, the strongest innate immune signal of danger is something called pyroptosis, which is an inflammatory cell death that's triggered by a set of these sensors in the cytosol called inflammasomes. So, as I already told you, we need 3 signals to really activate both cytotoxicity and memory, which are both properties that you want for anti-tumor immunity. You want to kill the tumor cell, and you want to have lifelong responses to the tumor. And so we wanted to know if we could induce danger signals in the tumors, could we enhance tumor immunity? And this sort of fiery death called pyroptosis that's sensed by sensing nucleic acids in the cytosol basically has several properties. It's mostly been studied in the antigen-presenting cells and macrophages and dendritic cells, but it can also exist in other cells. And basically, it's mediated by the recognition of DNA by either sensors that trigger interferons or sensors that trigger inflammatory cell death. And the inflammatory pathway involves activating a set of proteases called caspases, a subset of them that are activate inflammation. And when they do that, they cleave a molecule called gasdermin that can form pores in the cell membrane, which form this very inflammatory kind of cell death where, and process cytokines of the IL-1 family, which are very inflammatory. They're responsible for fever. So these cytokines and other small molecules in the cytosol get released, the cells die, and it causes the recruitment of immune cells to the site of damage. It enhances their activation, and it promotes their function into, say, killer cells. In humans, so this kind of cell death has mostly been studied in macrophages and dendritic cells and depends on one of the gasdermins called gasdermin D. But there are actually 5 gasdermins in humans, A through E, and any of them can be activated by cleavage at this linker region to cause this kind of inflammatory cell death, which is incredibly immunogenic. So this is just a cartoon of the inflammatory system in antigen-presenting cells. The sensor is called the inflammasome. When it senses a danger-associated molecular pattern, it recruits an adapter called ASC. It assembles into a large complex of about a micron in size that recruits the pro-inflammatory caspase-1, which needs to be activated by cleavage, bringing two of these molecules together. They auto-activate each other, they cleave each other, and then there are two main substrates, these inflammatory cytokines in the IL-1 family, as well as in these myeloid cells, gasdermin D. And the gasdermins have two domains, an N-terminal domain, which is the active domain, and a C-terminal domain that inhibits it. And when the inflammatory caspase cuts it in the linker, the inhibition is removed and the N-terminal domain goes to the cell membrane and forms a large pore. And the IL-1 family cytokines, these incredibly inflammatory mediators, don't have a secretion signal. They need to be processed, which is done by this caspase. And then their secretion depends on formation of these large pores. So we got involved in this about 7 years ago when Jing in my lab, so the year before this, Vishwa Dixit and Feng Xiao had identified gasdermin D as the important mediator of cell death and sepsis through genetic screens. But nobody knew how gasdermin D worked. And in fact, at the time, there were only maybe 5 papers in the world on the gasdermins, and nothing was known about them, essentially. So Jing fed the N-terminal domain of gasdermin D into a homology program. There wasn't any amino acid homology whatsoever, but this program suggested that the structure of the N-terminus of gasdermin D might resembled the killer lymphocyte pore-forming protein perforin. So we figured out that, or we suspected that gasdermin D might form pores in membranes. And in fact, what Jing did was he first looked at whether the N-terminal domain of gasdermin bound to phospholipids by applying either the N-terminal domain, the C-terminal domain, or the full-length protein to lipid strips that were dotted with different phospholipids. And he found that the N-terminal domain only bound to lipids. It bound to phospholipids that are acidic and on the inner leaflet of the cell membrane only. So these pores actually form from within the cell. If you took the active gasdermin D and added it to mammalian cells, nothing would happen. And it also bound to cardiolipin, which is a lipid on bacteria. So and basically what he found was this active form of gasdermin actually killed bacteria. So the things that stimulated this immune response were actually killed. And then Xinbin in Hao Wu's lab, who's my collaborator in all this work, showed that the N-terminal domain that was produced by an inflammatory caspase incubating it with the full-length gasdermin, neither the full-length nor the caspase did anything to membranes, but the N-terminal, the cleave formed these large pores of about several hundred angstroms in size. So they're big enough for a lot of small molecules like the IL-1 cytokines to pass through. And Feng Xiao's lab had a paper at the same time as ours showing basically a lot of the same things. And then Hao's lab went on to solve the structure of the pore, and it's just so beautiful, I can't resist showing it to you. Basically, Shiyu and also Jinbin for Gasdermin A showed that there was a large pre-pore that we identified of the molecule that looked like the uncleaved version, a sort of globular protein. But he also identified a large pore where there's this dramatic shift in the structure of each monomer into the hand-like structure, where the fingers form beta sheets and the pore is like a beta barrel that digs into the membrane and forms a pore. And this is just showing how it evolves into the pore. And it turned out that the pore had this acidic rim on the cytosolic side, and that the very inflammatory IL-1 family cytokines are acidic on their outside. So they were prevented from going through, and it's only after they were processed by caspase-1 that they actually were secreted. So we're getting to cancer. I just wanted to make sure that you had the background because this is really a new field and very few people actually know about it. So Feng Xiao and Ahmed Al-Nemri found that Gasdermin E, which is one of the other gasdermins, can be activated during apoptosis. So by caspase-3, which is a non-inflammatory caspase that triggers a non-inflammatory cell death called apoptosis in most cells. But if cells express Gasdermin E, which is actually the most ancient of the gasdermins, it's so ancient that there are homologs in bacteria, in fungi, and basically all cells. So it's actually probably part of the innate immune recognition of danger throughout evolution. That if cells express Gasdermin E, this non-inflammatory apoptotic caspase causes pyroptosis, inflammatory cell death. And we wanted to know, could pyroptosis be an important danger signal in cancer? And we noticed that Gasdermin E is not expressed in most tumors. So maybe it's immune edited to be suppressed. And in fact, in most tumors, the promoter is hypermethylated, so it's not expressed. So Zhibin and Ying in my lab sort of attacked this problem. They took tumor cells like B16 melanoma and treated them with a chemical called raptinol, which induces apoptosis. So in the wild type B16 or in control B16 cells, if you add raptinol, the cells undergo classical apoptosis where they shrink and they have these tiny little blebs. But in B16 cells where you overexpress Gasdermin E, what happens is that the cells undergo this funny kind of cell death where they form these giant balloons and their membrane gets damaged. So there's a green dye in the medium here, and the membrane damage is signaled because the green dye is taken up only in the pyroptotic cells. In the classical apoptotic cells, the membrane isn't damaged until hours later, and the macrophages in the body recognize apoptotic cells, phagocytose them, and eliminate them very rapidly. So classical apoptosis in cells that don't express gasdermin E is non-inflammatory, whereas in this situation, these cells are actually spilling out all kinds of inflammatory mediators and causing pyroptosis. And basically what we found is that expression of gasdermin E didn't affect, this is in B16, it didn't affect how much cell death there was. It only determined whether the membrane became permeable, whether it took up the small dye called cytox green or released large molecules like LDH into the medium. So basically what Zhibin and Ying found was if we overexpressed gasdermin E in a tumor in red, the tumor didn't grow or barely grew. If we expressed a form of gasdermin E where the site that caspase-3 cuts in that linker region is mutated, the tumor grew just the same as the wild type. And when we looked at this killer cells that were infiltrating the tumor, we found that the T cells, there were both more of them and that they also express the cytotoxic molecules, granzymes, and perforin that are needed to kill. The NK cells in the tumor also became activated, and they also, the T cells also much more efficiently secreted interferon gamma, which activates antigen-presenting cells, and TNF alpha. And then what we did is we took tumor B16 cells that were either treated with to express nothing, an empty vector, or to overexpress gasdermin E, and we tried a vaccine model. So our theory was that the gasdermin E expressing tumor cells would be very immunogenic, that they'd induce an immune response, and that they'd be able to withstand the challenge with the wild type B16 cells. So what we did is we vaccinated mice on one side, one flank, with the B16 that didn't express gasdermin E or did. And then we came back 10 days later with wild type B16 cells. And then we looked at tumor growth and whether there was activation of caspase-3 and gasdermin E. So here you can see the activated caspase-3 and gasdermin E only in the cells that were expressing gasdermin E. So we don't know how this gets triggered. What we think is that in a tumor, as it's growing, parts of the tumor don't have enough nutrients or oxygen, they spontaneously undergo cell death, and that's enough cell death to recruit all these immune cells to the tumor and very well control it. So in mice that were vaccinated with the wild type B16, most of the tumors grew, as you can see here. But in mice that were vaccinated with B16 expressing this inflammatory mediator, most of the tumors were suppressed. So what that suggested is if you could somehow induce a danger signal in the tumor, you would be able to control the tumor, or you might be able to potentiate other kinds of immune therapy. And this protection by gasdermin E turned out to depend on killer lymphocytes. So if we grew the melanoma in wild type mice, again, we got suppression of tumor growth. But if we took mice that were profoundly immunodeficient, that didn't have NK cells or T cells or B cells, then there was no significant suppression. And similarly, if we grew the tumors in mice that lacked perforin, the pore-forming protein that's required for killing, the tumors were no longer suppressed. And then what Gibbon and Ying discovered was that actually the killer lymphocytes themselves can induce pyroptosis in the tumor. So here we're looking at another tumor cell line, HeLa, and that either express Gesturman E or don't. And we're adding NK cells to the tumors that are the big cells that are adherent here. And if they express Gesturman E, we again see these giant balloons that are pathognomonic for pyroptotic cells and uptake of cytoxine, whereas in the Gesturman E negative tumor, we get the shrinkage and classical apoptosis. And what we found is that Gesturman, that granzyme B, which is one of the main proteases in killer cells, actually cleaves Gesturman E directly, and it cleaves it at the same site as caspase-3. So this is sort of background, but I'm taking up too much time, which suggests that if you could induce pyroptosis in a tumor cell, you could get immune control. And that killer cells actually can induce a danger signal if the tumor expresses Gesturman. So Gesturman E isn't expressed in most tumors because its promoter's hypermethylated. It can be induced by interferon gamma, which is one of the innate immune danger signals via the IRF2 transcription factor. So we thought, we wanted to know, could Gesturman E be repressed during tumor immunoediting? And what about other danger signals, innate immune pathways in tumors? So remember I told you people haven't looked at what genes get repressed because nobody can get hold of these early tumors. And so what we did is we took advantage of a genetically engineered mouse model of breast cancer, of aggressive breast cancer, in which there's an oncogenic version of HER2 that's induced by doxycycline. And in these mice, within a month, they almost virtually all develop tumors. So it's a very rapid and aggressive metastatic breast cancer model. And the tumors also are induced with doxycycline to express GFP. So we could isolate very early tumors before they would be visible to the naked eye. The tumors were driven by a mouse memory tumor virus, so they were in the breast tissue of the mice. And then what we did is we did single cell RNA-seq to compare what genes were expressed in tumors that were induced a week ago, so very, very early in tumor genesis versus big tumors at a month. And we analyzed 25,000 cells, and we're able to capture immune cells, fibroblasts, and epithelial cells in the tumor. There were differences in the distribution in early tumors versus late. And we could identify many subsets of T cells and epithelial cells within the tumor. And what we decided we took the most undifferentiated cells in magenta here and defined them as tumors. Some of these other ones, because they expressed EpCAM, which is a tumor antigen in breast cancer, GFP, which says that they were induced in our model, and they had they were very undifferentiated. And so we looked at what were the differentially expressed genes in the early versus late tumors. And what you can see is there were over 200 genes that were downregulated in the late tumors and about 100 genes that were upregulated. And when you look at the genes here, if you were an immunologist, you would see that these are all genes, practically every single one of them linked to innate immunity. And when we looked at gene ontology terms that were associated with these 200 odd genes, the pathways that were overrepresented were, here we have, in order, we sort of clustered them according to function, to genes involved in type 1 interferons, type 2 interferons, responses to virus, adaptive immune response, antigen presentation, and innate immunity more generally. And that's just shown here. The innate immune genes, adaptive immune genes, and there's only one little pathway of metabolism. So when I did this initially, I thought we're mostly going to get changes in oncogenes and tumor suppressors, but that's not what we found. This is really immune modulation during tumor editing. And this is just another way of looking at the interaction network of these enriched GO terms that were downregulated in the late tumors. And we put them as adaptive, innate, and metabolism. And metabolism can also be related to immunity as well. And then we could also look at the lymphocytes in the tumor and see how they evolved. And what we found is that the T cells that expanded in the late tumors were actually the exhausted T cells. So we can actually capture the immune, the loss of immune recognition and responsiveness within the immune cells. And if you look here at the genes in the T cells and NK cells that significantly changed, you see that in the late tumors, they lose expression of the molecules to kill an interferon gamma. They gain expression of the checkpoint inhibitors, and they lose expression of the chemokines that recruit immune cells to the tumor. And there are other interesting genes that are modulated there. And then we use the cell chat software to see what happened to the communication between, so looking at receptors and their ligands on all these different populations within the tumor, we saw that in the late tumor, they lost communications between the epithelial cells and the immune cells in the tumor. And when we looked at what was lost, it was co-stimulation in adaptive of antigen recognition and various cytokine and like interferon and IL-2 responses in the tumor. So I just highlighted here the communication between NK cells and the tumor cells that gets completely lost here in the late tumor. And light is a TNF receptor that pathway is also lost between the CD8 cells, the memory CD8 cells, and the tumor. So basically, I'm going to go really fast. The question was, so what we found is that during immune editing, the tumor loses the ability to be recognized by T cells. There were fewer T cells in the tumor, and the T cells that are there become exhausted. And the idea is, could we reverse the editing to make the tumor immunogenic again. And so since gene repression during tumor editing is mostly epigenetic, we decided we would look at what would happen if we used an inhibitor of DNA methyltransferases, since DNA methylation is one of the key features of repressed genes. And this is in the GEM model. If we added decitabine, which is a DNA methyltransferase inhibitor, to tumors that were already formed, and we chose, the tumors that were treated with decitabine were actually larger than the tumors that weren't. The tumors really regress. And this is like an incredibly aggressive tumor. And GEM models historically are completely resistant to immune therapy. And these are just the individual tumors. And then when we looked at the tumor infiltrating lymphocytes, we found that both the numbers of CD8 cells, CD4 cells, and NK cells increased in the mice treated with decitabine. In some of these subsets, like NK cells, there was more of the effector molecules, more cytokine production, and importantly, there was restoration of T cell memory. And then when we looked at the suppressive, this population of dendritic cells are the effective antigen presenting cells in the tumor. They increased, but the suppressor cells, the myeloid-derived suppressor cells, or the tumor-associated macrophages, really were lower in the tumor. Then we went on. I'm going to go really fast because I want to give you some time to ask some questions. Looking at a particular very aggressive breast cancer, triple-negative breast cancer cell line 4T1, we can see that decitabine has a dose-dependent suppression of the tumor. Again, in this tumor, there are more CD8 T cells, more memory T cells. They're producing more interferon gamma and TNF, seen here. And they have more functional antigen-presenting cells and less suppressor cells. Then when we looked at the genes that get de-repressed by decitabine, we saw the same kinds of innate immune genes, adaptive immune genes, and one population of meiotic genes. Not sure why, but probably because the tumor is very poorly differentiated. And when we looked at the overlap, first between the genes that were repressed in the tumor during development or upregulated after decitabine, there was a highly significant enrichment, and 2/3 of these genes in the enrichment are interferon-stimulated genes. And we see the same kinds of patent go terms that are enriched, either in this subset of genes or even in the ones that are not overlapping. So basically, the main genes that are repressed in the tumor are immune genes. And when we look specifically at a panel of innate immune genes that participate in this inflammatory cell death, I talked about another immunogenic cell death called necroptosis, the inflammatory cytokines, and the pathway that mediates type 1 interferon response. We found that in general, they were all upregulated. This is a heat map of the genes that we selected sort of before we looked at the data. And the same genes were also upregulated in vivo by treatment of decitabine. And if we have a method of delivering, of knocking down genes in tumors in vivo. with aptamer-delivered siRNAs. So we knocked down the DNA methyltransferases expressed in this tumor. And again, we got the induction of the same set of genes. Although it turned out that decitabine was somewhat better than knocking down the DNA methyltransferases, probably, and the main difference was there was more memory with decitabine. So the decitabine was acting not only on the tumor, but also on the T cells and promoting the memory response. And this memory, the effect of decitabine depended on killer cells. In perforin deficient mice, it was gone. We looked at it on several other tumors, including melanoma. We found the same suppression and immune activation. But interestingly, what we found is if we knocked out type 1 interferons, which was the top pathway, we still got pretty good tumor suppression. So not as good as here, but virtually the same. And so we did, we found that the decitabine actually induced not only the gasdermin pathway shown here, but also necroptosis, which is mediated by phosphorylation of this protein here. But what we found is if we knocked out different pathways in the tumors were still suppressed, not as well, and often not significantly, if we knocked out gasdermin E, or kinase 3, or the IRF pathway. And what we think is that that if you have any danger signal, any of these innate immune pathways, it's going to help you control the tumor. That knocking down just one of them, and that's why during tumor editing, they're all being suppressed. So this is the end. I'm sorry I went a little too long, but we found that we could actually study tumor editing using these genetically engineered mice, that we could identify changes in the tumor as well as what was happening in the infiltrating immune cells, and that we could detect the appearance of T cell exhaustion early on, and that all the major innate immune pathways were suppressed, and that reactivating even only one of them in the tumor could contribute to immune protection. And I didn't show you this, but we can add decitabine with anti-PD-1 and get synergy. So in tumors that don't respond to checkpoint blockade, we see better protection. And so if we go back to Ira Mellman's model of tumor immunity, what I'd like to suggest is that if you can get the tumor to signal that it's something dangerous, you'll have a much better immune control. And these are the people who did the work. Jing started this study of inflammation in my lab. Zhibin and Ying did the study with the gasdermin E, and Ying is the person who really led the work on that I just told you at the end. And all of this was done with amazing collaboration with Hao and the people in her lab. And the bioinformatics for the last study was done by Poria and Winston Hyde's group. Thank you. So do we have time for a few questions?
Questions, and people by Zoom can write down their questions and I can read them.
Don't be shy.
So there is one by Zoom, and that's by Daniela Goretti. She asks, has the wall immunological synapse been characterized in vivo in tissue with tumor cells?
No, I don't think so. I mean, no, I can't think of, I mean, I don't know the whole literature. It's enormous. But I can't think off the top of my head of a study where they actually look at the immune synapse. We developed a method of actually looking at immune synapses in cells that are isolated. We can focus on conjugates of T cells or potentially NK cells with tumor cells. And we were able, in the early days when people developed imaging flow cytometry, to actually look at the sequence of events that occur in the synapse. So it's possible. We can, one thing we can do, which I didn't show, is we can look at pyroptosis in vivo. So, and we've done that in this system. So basically what we can do is inject propitium iodide into the mice bearing tumors, and 10 minutes later, we sack the mice, isolate the tumors, and we can see dramatic increases in pyroptosis in mice treated with decitabine or with other agonists of pyroptosis. So we can actually see this. Nobody, it's almost impossible to see apoptosis, but we can see pyroptosis in vivo.
Oh, thank you, Judy, for the fantastic talk. I was wondering, the immune editing is a general mechanism, or is for all type of tumors, or is it specific?
That's the theory. So we've treated mice bearing different kinds of tumors with decitabine. We see the same response. We just got, there's a fabulous GEM model for colorectal cancer, which was produced by this guy, Edward Batel in Spain, where he takes the mutations that are, the sequential mutations in colon cancer, APC, 4 mutations. And those mice, again, they have a very rapid and aggressive colon cancer that's not responsive to checkpoint inhibition. And we're going to repeat the studies in that model. And I've talked to a cancer immunologist in Chicago who studies ovarian cancer. He has a GEM model. So I'm hoping that we'll be able to study what happens in other tumors. Just as well.
Yeah, if it's a general mechanism, really is a great discovery. And my second question is, what signal triggers the immune editing?
Well, I think it's natural selection. So if you think of the tumor, it has to, it wants to grow. And if it's going to grow, it has to somehow avoid being killed by the immune system. So I just think it's natural selection. Every time, the tumor's dividing, so there are many tumor cells. Some of them will have different, levels of repression, and the ones that survive and then can multiply without limit because the immune system ignores them.
Thank you.
By Zoom, Dr. Mario Cruz asked, what about the role of cancer means in pyroptosis in lymphoid cells?
In lymphocytes?
Yeah.
So lymphocytes, naive lymphocytes, resting lymphocytes, don't express any of the gastrodermins. So they're not but susceptible to this pathway. However, what we found is if we treat lymphocytes, we activate them, they then induce all the gasdermins very strongly. So I think that in vivo, in situations where there's inflammation, you're going to have lymphocytes that are going to be susceptible to pyroptosis. And, the truth is, this is a very new field. So we don't know a lot about what regulates gasdermin expression, but I told you that interferon gamma will upregulate it. So you can think of situations where there's macrophage activation, and I bet there's going to be a lot of pyroptosis going on.
Okay, we have another question by Dr. Enrique Renaud. He asks, could the Citabine be used before having cancer detected as a vaccine against cells that begin to be altered and recognized by the immune system?
Yes, so one of my colleagues, John Kagan, and in some situations, when you activate gasdermins, the cells don't die. They repair the membrane damage, but they still produce a lot of cytokines. And what he found, and that he described that in macrophages and dendritic cells. And he had a paper, I forget what journal it was in, a couple of years ago, in which he showed that these dendritic cells that are activated to, he called it hyperactivation, because they actually secrete much more cytokines. They're much better antigen presenting cells. And if he incubated those dendritic cells with tumor antigens, they made cold tumors hot. So he could take a tumor like B16, which is pretty cold, and make it immunogenic by a tumor vaccine.
Yeah. Can this logic of immune surveillance and immune editing be also applied both during the epithelial mesenchymal transition, like in order to avoid the entire transition before even the tumor finished to grow?
I don't know. I think it might be possible to test that. I mean, we showed that you could induce EMT. Well, I think you'd have to really think hard about how you're gonna induce EMT, and how you're going to get to look at the cells that are undergoing the transition. But I think it would be possible, and I think it's, those cells are really important in cancer. So definitely worth thinking about.
So we have another question again by Dr. Mario Cruz. He asks, is it possible that gasdermins are overactivated in inflammatory diseases?
That they're always activated?
Yeah, overactivated.
Yeah, so there's, as I said, this field, the gasdermins were only sort of identified as important in inflammation 7 years ago. But already there are many studies in all kinds of models that implicate gasdermin D, especially in the pathology. And they include studies of cardiovascular disease, obesity, Alzheimer's disease. There's a paper that we're involved in that was just accepted about ALS. And in terms of there are autoinflammatory genetic diseases where the inflammasomes are activated, where the gasdermins are critical. Sepsis is a really important one. SARS-CoV-2, we had a paper this past year in Nature about the importance of inflammasomes in severe COVID, which is an inflammatory disease. So this is a new field, but I personally think that this pathway is involved in almost almost every pathological disease has an inflammatory component that either that exacerbates it or maybe even be critical in initiating it. And this is really the most inflammatory pathway that exists. So, so we've developed, we identified molecules that can inhibit Gerstermann pores. We haven't published this yet, a molecule that can activate it, which you might want to do for cancer. And I think the other innate immune pathways are also important too. So
the last question in Zoom is by Dr. Aislinn Perez. She mentioned it is understood that methylation has a large effect in lowering the immune response. Did the tumors continue to grow more than 30 days? That's the first question.
Well, we had to sacrifice. So we basically sacrificed all the mice when the control tumors were too big for our animals. So we never saw if the tumors started regrowing. And
yes, and the second question is, does the immune response recover after DAC treatment or does it decrease again?
We haven't looked. We just basically all we've done is most of what we've done you've seen there. I think we have to carry things out further and see what happens. But the fact that checkpoint is a curative in most situations, and that resistance doesn't develop, one could be hopeful that if once you trigger a really powerful immune response, that it will persist.
Another question? No? If not, thank you again, Judy, very much for your nice presentation.